Hi, I'm Audrey Jordan, and today we're going to discuss the issues with Okapia johnsoni detection. The okapi is an endangered African ungulate. It's also known as the forest giraffe. And the issue here is that they're very hard to detect. They're endemic to the central and northeastern Democratic Republic of Congo rainforest. So this means that their habitat is very, very dense, remote, and hard to get to. They're also endangered, so the numbers are very, very small. The species is not abundant. They're very elusive, and there has been no established method of detection. So the methods primarily used are camera trap, scat identification, and they're just now starting to use eDNA. And no one of these methods has been seen to be exclusively highly successful. And this picture here is actually a great camera trap photo of the okapi, and we can see the dense vegetation that surrounds it. I also included this map so we could get a better idea of the habitat of the okapi and how it makes the detection so difficult. The red spots are going to be the occurrence of okapi dead or alive. And we can see that they're concentrated and fragmented throughout rainforest areas in the DRC. The first method that's primarily used is camera traps. This consists of placing a game camera on a tree, and then the camera will be triggered to take a photo when an animal passes or vegetation gets in the way. And the pros of this is it can be deployed for a very long time. You have a long time span of collecting data. It's passive, meaning it's not going to negatively affect the okapi, and it's relatively low cost for the amount of data you could potentially be collecting and the time span that it would be out there collecting data. Some of the cons of this method is that the okapi habitat is very dense vegetation, the cameras can't see through vegetation, and as I mentioned earlier, the camera will be triggered by anything passing in front of the sensor, so that could incur a very long data processing time. And the next method is going to be scat identification. This method is employed by walking with transects and counting the dung on the transect, or it can be used as an indices to try to estimate the density of the okapi. It is also passive and great for not impacting the okapi. It's cost effective using little to no technology and manpower. And the scat identification could lead to collection, which could give us some genetic information about the okapi, which is great. But some of the cons is that it's very difficult to identify okapi scat compared to bongo scat. They often get confused. And again, the remote dense habitat is very hard to traverse. So you'd be wasting a lot of time navigating between transects, and some places just are not possible to get to. The last method that's used is eDNA. This stands for environmental DNA. We're taking DNA left in the environment, could be in soil, water, and a new technique is actually trying to use air samples, which has been seen successful in zoos. And then it will establish occurrence of the species in the area. And a pro of this is, again, it's passive. It will not be disturbing the okapi population negatively in any way. The animals also don't physically have to be present, which is great for the okapi because they're so elusive, very hard to find. And it's relatively inexpensive for the amount of information you're going to be acquiring. And this is a very short period of time. It's not a long study. You go out, get a sample, bring it back to the lab, and analyze the sample. Some of the cons of this, though, is it's difficult to detect animals in small densities, which the okapi is, in very small densities, and it's best used for a snapshot of biodiversity. My recommendation would be scat detection dogs. It would decrease the time for humans to traverse, and it's cost effective. There would be guaranteed correct identification if they train the dog on the scent. And the cons would be the inclement weather of the rainforest would cut down on scenting accuracy. And again, traversing remote locations, even though it's a dog, is still going to be difficult. Here is the literature that I used, and thank you so much for watching my video. Leave a comment down below and ask me any questions that you have. Thank you.